Hi all, welcome to the fourth in our series of the Go with the Flow expert webinar series. We're just going to wait maybe a minute or two here to allow some folks to join us, and then we'll just get started. So just be patient for 30 seconds or a minute, and then we'll get started. Thanks. Hi, everyone. Welcome to our fourth in the Go with the Flow expert webinar series. I just want to wait a couple more minutes here, maybe a minute or two before we get started to allow some more attendees to join us. So let's be patient for, say, a minute or two, and then we'll get started. Hi, everyone. Welcome to the fourth and final talk of our Go with the Flow expert webinar series this month of June. Just to reiterate, for those who have come to some of them, we'll still have one more next week with our panel discussion with Tony Hunt, our CEO of Repligen. I hope that everyone can come and ask a couple questions to Tony, give him a hard time. That'd be fun. In any case, today I'd like to welcome Raf Dedier. He's the Associate Director of Advanced Technology at Janssen in Schaffhausen, Switzerland. Raf has had a wealth of experience with both the FlowVPE and the FlowVPX, primarily focused on the dilution process and the drug product side of the business. And he's here to talk about his experience with that. So without further ado, I'd like to introduce Raf, and he can take it from here. Thanks, Raf, and I look forward to hearing what you got.
Thanks, Ramsey. And good morning, good afternoon, everyone, also from my side, and thank you for joining the webinar organized by Repligen. And as Ramsey highlighted, today I will be talking more specifically about our experiences, how we've been using the FlowVPE system in over the last couple of years in our drug product manufacturing processes, and then more specifically how we've been using it in our dilution process step. So I think the presentation, if all goes well, will take around 30 to 40 minutes. So that means that at the end of the presentation, we'll have plenty of time for Q&A. So if you have any questions, please hold until the end, and you can ask them via the chat, and then we can discuss them at the end of the presentation. Good. Then, with that said, this presentation is going to focus a bit more on why protein concentration is so important or so critical to our biologics drug product fill finish manufacturing process. And so how we're actually utilizing the FlowVPE instrument to support our business need. I'm also going to talk a bit more about how we have gone about implementing the FlowVPE tool into our GMP manufacturing processes, because that's also, let's say, more than simply plug and play an analytical instrument into your manufacturing process. And then lastly, I'll just give some pointers on how we are actually moving from protein concentration monitoring to advanced process control. Good. So let me just start by introducing you to the world of drug product fill finish, because I do know that especially around FlowVPE or within the biologics, say PAT world, there's a big emphasis on the drug substance side, both upstream as well as downstream, and very few is often taking place in the drug product world. And one of the reasons why that, in my opinion, is because of the, let's say, the high demand for sterility of all of the processes that we have in our manufacturing facilities. And I think also the goal of this presentation will be able to, or I hope to be able to give you some light on how we've gone about some of those concerns, and we're successful in implementing the FlowVPE into our manufacturing processes. So on this slide specifically, you can just see a schematic overview of what the different unit operations are that typically take place in the biologics fill finish process. So we generally start from a drug substance container, which contains a highly concentrated solution of the protein that we would like to see ended up in our final product. And then together with some other excipients, salts, sugars, let's say acid or alkaline components, are then, let's say, mixed into what we call then the final formulation or the final compounded solution. That specific solution is then subsequently transferred into a filling isolator, which is really the aseptic sterile core, where we then have successive filtration steps to render the solutions fully sterile, and then that solution is being filled with a pump system into vials or syringes or any other type of primary container that you desire for a specific product. And then once that's completed, then the vial or syringe then goes off to optical inspection, packaging, and all of the other more downstream processes before it ends up at the patient. Now, protein concentration actually is something that typically is measured several times throughout our manufacturing process, and it's one of many, but I would say it's probably one of the most critical quality attributes that we have, really because it determines the final dosage that the patient receives during treatment. So to that extent, I said across multiple time points, where that would be on incoming materials, where that would be as part of the formulation step, or even on the final vial, we do pull samples to determine the protein concentration. And generally that goes as in many other manufacturing processes, where we take a sample, that sample goes to a laboratory, where they then do a destructive test on the sample to determine the protein concentration. So what we actually set out to do, I think it was around 2016, that we wanted to evaluate whether there was a possibility to perform any of these tests online. And our main focus initially was really to speed up the testing, right? So generally we pull the samples, and then the sample goes to another building for analysis. And in the meanwhile, our operators are waiting for the results. So we just wanted to find a solution in order to speed up that testing and reduce the lead time. But as you'll see in some of the case study examples that I will highlight in the next slides, we've gone about utilizing the FlowVPE for many other purposes rather than just lead time reduction. So initially it all started around what tool that we initially wanted to implement for determining protein concentration. And I will not spend too much time on highlighting what FlowVPE is, because I assume that most people, or even all attending this webinar, do know what the FlowVPE system exactly is. What I do want to highlight here is just the reasons why we, from a drug product manufacturing side, ultimately ended up selecting FlowVPE over other technologies that we also evaluated. And I'll there are a couple of more details that I'll talk about at a later point in this presentation, but just from a high-level perspective, I think there were two very main important reasons, and one is that we do have a very broad variety in concentrations that we typically see as part of drug product, right? So as said, we have highly concentrated solutions coming into our facility, and sometimes very low diluted concentrations exiting our facility. So we do see a whole breadth of concentrations in our processes, and the tool such as FlowVPE is really capable exactly for determining that with one specific piece of equipment. And then I think the other reason was also that it's still a UV-based method, and because of that reason, it utilizes all of the product information you already know, in contrast to other technologies like near-infrared, for instance, that would require you to build a specific model that would be able to extract the quality attribute that you're looking for. Now, in the next couple of slides, I said I'll just highlight some of the initial case studies that we've done whenever we acquired then the FlowVPE in our facility, and then we started implementing it into our process. And just I'll talk very briefly on just some of the learnings that we've done before I will talk exactly on the implementation process. So one of the first runs that we did with the FlowVPE, we started to look at our filtration process. So you can imagine you have a tank that has a certain product with a specific protein concentration, and you're sending that through a depth filter that would really filter out all of the particles and microorganisms, and then we would monitor basically the output of that filter using the FlowVPE. And so one of the things that you actually can see from this graph, and it looks like a very static graph. So here we plotted just let's say the protein concentration monitored by FlowVPE versus the amount of volume that was sent through the filter. And I think one of the first learnings that we got from this application was just let's say the velocity or the speed at which we can get this data. So looking, for instance, how equally spaced are the individual data points, we are indeed talking about rather seconds, right, to generate one specific data point, which is something that would allow you to collect a very high amount of data as part of your process. Now, why I mean that this is very important has everything to do with some of the effects that we see generally during filtration, and that has everything to do with filter absorption. So what I'm now overlaying on that same graph is just the target limit as well as the acceptance criteria that we have for this specific product, which has a target concentration of around 5 milligrams per milliliters. And then with the red bars, you do see what the acceptance criteria are. And I think what is very critical here to identify is that with the FlowVPE tool, we were able to start seeing certain effects that we were not able to see beforehand unless we were doing very extended sampling. And the effect that I'm talking about really is that initial, let's say, small dip in the protein concentration that we see whenever we're starting the filtration process. And you do see that kind of levels off and reaches the final target concentration after about, let's say in this case, around 60 to 80 mils of volume sent through that filler. So, I mean, it's a very small effect that we've seen here, but the fact that we were able to capture data at such a high speed and with such an accuracy and resolution, that was really, let's say, for us, a starting point to identify what we could do with the FlowVPE moving further. And actually moving further, what we did, we repeated that same experiment, but we utilized different filters. And more specifically, we tried out the different filters that we use across all of our different manufacturing scales. So let's say the initial curve that you saw on the previous slide, that was a typical filter that we use normally as part of our development studies. The icons that are green, that represents a filter that we use as part of our clinical manufacturing activities. And then the light blue one, the graph on the bottom or the curve on the bottom, that's data representing a filtration process of a commercial filtration step. And so what we do see is that with increasing filter size and with increasing filter membrane size, we do see that effect of, let's say, filter adsorption taking place at the beginning of the filtration step is worsening, let's say, significantly, where for those lab development studies, you might not even have noticed that there was a specific protein absorption taking place. But then for, let's say, larger filters, you do see that the effect is becoming more prevalent. And let's say this is only just the result coming from one filter, while typically as part of our drug product manufacturing process, we do have redundant filtration taking place. So you could even consider that actually the effect that we're seeing here is almost even doubled in the case of a redundant filtration. So I think what this data for me just represents really, again, that the FlowVPE was really for us a tool that would allow to see certain events that we were not able to see initially, but also just the fact that a lot of studies happen on the lab scale, and sometimes we might not be able to see some certain effects because of the just because of the size that we of all the components that we're using at the lab scale. And because of that reason, having a tool like a FlowVPE, which is really something you can put directly into your manufacturing process, sometimes might even help to reduce the extent of the laboratory testing you need to do, but very often also just yields data that is superior to the data that you might generate on a laboratory setup. Good. And then maybe just a last aspect to the filtration study that we've done, and I'll not go into the details on this slide specifically, also because this data has not been generated by me specifically, but by our drug product development group. But again, here, the fact that you have that sheer amount of data coming off one tool allows you to do much more, which otherwise might not be possible with the traditional sampling approaches. So in this case, what we did was we used all of that data and we put it into a scientific model to really characterize the protein absorption, which is then something you can also use to perform, let's say, virtual experiments that you don't want to repeat, or you can't repeat in an actual setup because you have no material available. So I think an online tool such as FlowVPE and modeling sometimes go very nicely hand in hand in order to generate a lot of data that otherwise might be very difficult to obtain. Good. A second application that we have evaluated the usage of the FlowVPE was actually the filling process, right? So once you have your solution that has passed through the different filters, you're starting to then fill that into the primary container. And what we did there is we really monitored the protein concentration right during the process when you're filling a solution into the vial or the syringe. So if the previous graph was not boring enough, this one for sure is, because basically over a period of around 6 hours in this case, we have just monitored the protein concentration, and there is almost nothing to, or there is actually nothing to see in this in this graph where we have really constant reading as function of time. But let's say, even if this is a relatively boring graph, I think it is very important to showcase what can be, well, for me, two things, right? So one is that also here again, the FlowVPE is something that really can be used if we're talking about all of these initiatives such as real-time release, for instance. Having a FlowVPE installed right during your filling process allows you to determine, for instance, what's the exact protein concentration of your solution entering into each individual vial. So if we're talking about maximizing product quality, instead of having a batch that maybe contains 20,000 vials or syringes and you sample 3 out of those to determine with a destructive test your protein concentration, this would allow you to have really 100% of monitoring. So for each individual vial, you do know what's the content of that vial. And as said before, protein concentration is really one of the most critical quality attributes because it determines the dosage the patient receives. So if you do detect that there is, for instance, somewhere a small temporary reduction or an increase in protein concentration, you can simply cancel out that specific vial while still, let's say, having a normal continuation of the filling process and not having a bigger impact that could potentially jeopardize the whole batch if that same issue would have been detected as part of the QC release testing. And then I think a second aspect that is pretty important to highlight is just also the accuracy, right? So drug product being really the final step in the whole manufacturing process before the patient gets shipped to the patient, we do need to ensure that all of our attributes are within very tight specifications. And having now this example again of a product that has a 5 mg per ml target concentration, seeing now that data being collected over the period of 6 hours, that data is really able to very accurately, or the instrument is able to very accurately pinpoint exactly that concentration with such a high accuracy that allows really to be that the data can be used for quality release purposes as well, and not just as, let's say, a process development tool, such as for the filtration study that I talked about a couple of minutes ago. And then actually the third application I do want to talk about today, and I'll talk about that more throughout the presentation as well, is a dilution process, right? And the reason why I really want to spend a bit more time on the dilution process is that here we do see the usage of the FlowVPE not only for monitoring, but also, let's say, having more extended usage for instance, controlling the manufacturing process. Now, when we talk about a dilution process, typically or traditionally, that would be a 5-step process, where in step number 1, you are creating a formulation buffer which has all of the components except the protein. And then in step 2, that's where you actually take the protein as your raw material, so a highly concentrated solution. That's your starting raw material. And then you need to mix those two together in order to end up with your final formulated product that you will filter, and then you will fill into vials or syringes. So traditionally, how we do that is that in step number 3, we'll take an actual sample of the incoming raw material to determine, again here in the QC labs, what's the protein concentration, and then that data is being fed back to the manufacturing shop floor. And then what they'll typically do is an operator will take that data and then will perform a certain manual calculation to determine what's the weight of the buffer that needs to be added to that highly concentrated protein solution in order to end up with the final diluted drug product compound. And then an operator will typically do that, execute that process. And then at the end, and when the whole process has been completed, there will be another sampling performed in step number 5, where then a sample goes back to the QC lab to really confirm, okay, have we correctly executed that process, yes or no? If no, then is there any possibility that we can still save the batch? If it's over diluted, it's anyway end of the story. But if it's under diluted, we might be able to still recover some of the material. But if it's okay, then we can continue with the rest of the process. And during all of these, let's say, intermediate analyses, operators are obviously waiting for results. But what's also very important is that you see that as part of the whole manufacturing process, we do have multiple manual interventions, right? So we are sampling offline, we are analyzing by a human person offline for protein concentration. The data has been fed, will be fed back to the operator to determine with a simple manual calculation what to proceed next. So there are a lot of manual interactions. There are a lot of, let's say, potential errors, let's say human errors that could come up in this process. And what we have seen here specifically is that just implementing the FlowVPE as part of this whole process will eliminate to a large extent that issue. So what we are now typically doing as part of a dilution process is that this is still a 5-step process at this point, and I'll go to further optimization later on this presentation. But for this 5-step process, what we are really doing is we're replacing the QC lab with the online FlowVPE instrument. So instead of having to perform a certain sampling, the FlowVPE is giving us the reading at the moment we are having that raw material available to us, and that data is available within seconds rather than hours. And then with that data, an operator is then able to calculate and determine how to execute the process. And then once that's completed, also within seconds, we have again the data coming from the FlowVPE instrument to really give us the pass or fail whether this process has been executed correctly, yes or no. So from, let's say, from a benefit perspective, implementing FlowVPE as part of our dilution process has, on one hand, really allowed us to have a significant time reduction, because also here we're not waiting for the samples to go to the lab, data to come back, but we're having the data really on the shop floor. And then, as said as well, the fact that we're not just monitoring only the beginning of the process with an offline sample and only the end of the process, we're able to generate, let's say, every handful of seconds, a protein concentration value. So having that wealth of data throughout the whole dilution process allows you to also have a potential, let's say, prevention of execution failure. So if we do see that there was a human error that went, let's say, into that calculation of how much buffer needs to be added into the tank, then FlowVPE would actually see that we're maybe going much faster towards the target concentration, and we might potentially overshoot the target concentration, and we're over diluting the product. Having that real-time data available really is allowing to put, let's say, the operator in the better spots to make good decisions and fast decisions around execution of the batch and execution failures that might have occurred. And then maybe just the last comment is that the fact that now you're bringing that tool really into the hands of the operator is removing also the fact that you need to have very strict communication between the different departments. You have an operational shop floor, you have the QC department that's doing the testing. So imagine during a night shift where you need to coordinate whenever you're going to pull the sample and send it to the QC lab. What if there is nobody there that needs to support the testing? So by having this online tool directly available on the shop floor, this really, let's say, enables that operational flexibility because it's all in the hands of, let's say, one specific group. Good. So with that, kind of just want to wrap up the first part where I hope I've been able to just showcase to you that the FlowVPE system, how we've been using it in the, let's say, the drug product applications. We've been able to monitor basically almost every single unit operation step that we have. So the compounding or the formulation, the filtration step, the filling process. So we've seen value in utilizing the FlowVPE across the whole drug product biologics manufacturing process. The main benefits that we've identified so far really is that you have that real-time analysis of protein concentration, so you really can omit the let's say the offline sampling and the offline analysis. But I think a very important learning for us after these initial feasibility tests was really that we are indeed generating a lot of data that we might not have been able to gather previously. Either it was because there was a need for a lot of sampling and destructive testing, or sometimes it was just not possible to have such a resolution in time, for instance, to even capture the data with sampling approaches. So I think that was a very nice learning coming out of those feasibility tests. Good. Then in the second part of this presentation, I just want to give a bit more information on the actual implementation journey. So how did we now introduce the FlowVPE into our GMP manufacturing processes, and which steps really are crucial on that path. So before I do that, I just want to emphasize that I might be the one that's presenting today, but this is really a team effort, right? And you'll see that on the next couple of slides as well, that the input from all of these different groups and departments are really required to have a successful implementation of a PAT tool such as FlowVPE. So you have analytical development that will have to develop the method for the FlowVPE. You have drug product development that is doing a lot of the process development and is also looking at how to and where in the process to implement the tool. We have quality control, we have quality assurance. We have manufacturing science and technology, so really the people that are responsible for the operations and linking it to an actual facility. And then we have technical operations that are also required for, say, cleaning validation, sterility validation, all of these different activities. And so I'll go in the next couple of slides a bit more in detail on what are really the different actions that we have undertaken from, let's say, purchase of the instrument until the implementation and the usage in a GMP environment. Good. And so basically that's kind of represented on this slide. So we within the organization have a process that has been established around the implementation of PAT sensors such as the FlowVPE. And as said, it also highlights all of the interconnected activities that are required and who are the different groups that are responsible for executing those activities. So we have, let's say, a standard process where we might go from technology due diligence, then we'll proceed with activities until we have successfully completed online lab feasibility. Then we'll have a bunch of activities up to the point that we can start using the tool for process monitoring, and then another couple of activities until we can really use the data coming from the tool for IPC or release purposes, or even for extended usage such as controlling the manufacturing process. Good. So where it all starts, right? So first step was really technology due diligence. So a couple of years ago, we really looked at various technologies to determine online protein concentration, and I'm just highlighting here a couple of the let's say the methodologies that we've been using, and so most of them are just, let's say, somewhere in that wavelength band ranging from UV, NIR, mid-IR, or terahertz technology. And I think ultimately we did end up selecting for the VPE technology mainly because of three reasons, right? So I have highlighted already previous in the presentation that accuracy and precision is really something crucially important for our drug product manufacturing process. If we have a tool that might be incredibly fast, but it's maybe only plus minus 5% accurate, it might not be good enough for really might be good enough for monitoring, but it might not be good enough really for IPC or release testing. So the fact that we have collected the data with these early feasibility tests that I showed you before with such a high accuracy using the FlowVPE tool was a very big decision point on moving forward with this technology. Second specific reason was really around any potential background effects, right? So within drug product, we do see probably not as much as a drug substance, but we also see a lot of influences coming from, let's say, raw materials that have certain small differences, different batch varieties, where we also want to have a tool that is able to give us the attribute that we're looking for without having to compensate for a lot of these different background effects like pH changes, for instance, that will take place during our manufacturing process. So, and then the third one, as said, again, the fact that a FlowVPE tool is really something that doesn't require any additional information to what you already have available within your organization. So you have standardly a A280 method already established for protein concentration, and that's the information you need. And there's no need for building a Raman model or an NIR model and characterizing all of the potential variations you might see in the background, for instance, to determine your protein concentration. There is really, let's say, only one parameter you would need for determining accurately the protein concentration, and that's information you have traditionally available during your development process. Now, after we've then selected the FlowVPE, we started to, or let's say one of the elements that we generally very early on start to think about really is where do we see this tool implemented and how can we physically implement this tool as part of a manufacturing process. So there are a lot of so to say PAT sensors that are available, but sometimes the difficulty really lies in how do you get this tool interfaced with your existing manufacturing equipment. And so this is where I'm just highlighting here now in two examples where the design of the flow cell, including the actual components that make the flow cell, were allowing us to integrate the whole FlowVPE quite easily with our manufacturing process. So in this case, just starting maybe on the bottom, the filtration and the filling processes that I highlighted in the beginning of this presentation, since it's a flow cell and our filtration and filling processes are anyway continuous processes, a flow cell with then the provided, let's say, easy standard connectors allowed to integrate this flow cell directly into, let's say, standard tubing or standard pipes that we use during our manufacturing processes. If we now look at certain mixing studies or compounding or formulation steps that we use, those are typically batch processes. So those are the ones that take place into a closed environment like a compounding tank. And so whenever we wanted to then incorporate or include the FlowVPE during this testing, was really that we had to set up a recirculation loop specifically for that, right? So we had a loop where there was continuously material being extracted from the tank. That material was then sent through the flow cell, and then using a peristaltic pump in this case, we would then actually send the material back into the tank such that we would not have any loss of material, and then the material could go back into the process and could be used for further processing. And so the fact that we had that flow cell with very easy standard connectors allowed really us to easily set up such a recirculation loop. Now, so once, let's say, the initial due diligence and then the feasibility testing is completed, we then went on with the first online lab feasibility. And this is a picture from around 2016, where you can see also here what I just explained on the previous slide. We have a compounding tank. We have then actual recirculation loop with the FlowVPE instrument, with the laptop as well, and then a peristaltic pump to actually bring it back into the tank. And as you can see at this point, it really looks like a very much customized setup, so not at all ready for final production, but at least good enough to understand just the benefits and the drawbacks of the technology that we're evaluating. And so what we specifically did in this study was we just looked at mixing studies, let's say in utilizing various tank sizes, and this was in our clinical facility. And so once we basically have a technology that's now potential to be implemented, we've collected some initial data, we do have to go into that next part, which is really how do we bring it into a GMP facility. And I think one very crucial element that we had to do was to determine whether your change to the process that you're applying or the tool that you're integrating, whether that's in any way negatively impacting the quality or the stability of the drug product in case. So in this example, we have a FlowVPE, which is continuously sending out UV radiation to the product. We also have, in the case of a dilution process, we have this recirculation loop that's continuously going around, extracting material, sending it for analysis, and sending it back into the tank. So that's adding a specific stress, or potentially that's adding a specific stress to the product. And then on top of that, you're having all of these materials from the sensor and all of the recirculation loop or other parts that have potential leachables and extractables that might have a toxicological impact or toxicological effect on the patient. And so all of these elements need to be evaluated and need to be verified that, as said here, that the impact on the patient and the impact on the drug product quality and stability is not negatively impacted by the usage of a tool, as well as any of the auxiliary equipment needed to accommodate the tool, like the recirculation loop and the pump. So for this specifically, we typically set up a product dedicated study where we mimic the actual production scenario under worst conditions. So from a batch size perspective, from a recirculation loop speed perspective, measuring frequency perspective, and then we're just evaluating various quality attributes and stability indicators to determine whether we see certain degrading influences, yes or no. And that will generally help to determine also what's the boundary that you can use FlowVPE. So if you start to see after 10 hours of usage, as an example, you start to see certain stability indicating degradation, then you might want to limit, for instance, the usage of the PET tool to a short amount of time. And so this study we've completed for a handful of products now, and has really been able to exactly determine, and with a high assurance, tell us that the usage of the FlowVPE for the application that we were looking at was not negatively impacting the quality or the stability of the product. Then similar to an offline lab method, you still, let's say the FlowVPE is an analytical tool, and there needs to be a method validated or developed and validated in order to be able to use that data for GMP purposes. So we have for offline methods, we have very clear guidelines from ICHQ2 on how to validate the method, so whether that would be linearity, accuracy, precision, and all of those other parameters. But then now you're having an online tool, which also comes with certain dynamic aspects, right? So you have also here maybe potentially the effect of a flow rate that might be higher or lower that could impact the accuracy of the reading. And so also for this, the analytical method validation should reflect, let's say, the boundaries of that dynamic aspect of your PET tool, and those should also be incorporated as part of your method validation. And this approach we've also done, let's say, successfully for over 4 products to date, where we have been able to validate the FlowVPE for all of those methods, adhering to any of our, let's say, predefined acceptance criteria, whether that would be for IPC or for release purposes. And then last but certainly not least, right, is the whole cleaning and sterilization validation exercise. So you have a lot of parts coming from the sensor, as well as all of the auxiliary parts you would need to interface that sensor, and they need to be cleaned, they need to be sterilized. And there is no real one approach fits all, but this is also very highly dependent on how the tool is going to be used, whether that's part of the filling process or the compounding process, so after or before the sterile filtration. But in any case, there needs to be assurance that the cleaning processes are able to clean all of the residues from the sensor, and also that the sensor is able to withstand and survive the cleaning process. And then the same for the sterilization process, needs to be able to withstand multiple times for extended durations as well, the sterilization procedure. And that's really a very critical item to tick off, so to say, as well, right? So where in our case, the FlowVPE, we are cleaning out of place using a small parts washer instrument, where we have successfully completed the cleaning validation on multiple products, on all of the FlowVPE components, whether that would be the flow cell, the fibrate, the auxiliary connectors, and all of these, where we did swap and also test for organic carbon, bioburden, and endotoxin residues. And then from a sterilization perspective, we autoclave the FlowVPE components, and let's say for the dilution process, the compounding process application, we are there in low bioburden conditions, not in the aseptic core, but in low bioburden conditions. And that's where we also have done an assessment on whether the autoclave process, including the materials of all of the components from the FlowVPE, were able to be sterilized sufficiently, and also that was completed successfully. And so to date, we have been running repetitively the cleaning and the sterilization processes, and we do have always, prior to use of the FlowVPE, we're still doing a system suitability test to verify that none of the cleaning and sterilization processes negatively impacted the quality of the sensor parts. And to date, we have not seen any negative impact on that. And we're still in the process of collecting data to really see at which point we might have a failure of the components. But as said, we're still in the process of collecting that data. Good. And then maybe just to finish off the this second section as well, these are just some pictures over the period of 2016 after initial feasibility up to 2019, where we then really implemented the FlowVPE in our GMP facility. So on the left hand and the middle side, you can see the FlowVPE in our clinical facility. So on the left hand side, it was still, let's say, rather a customized setup, where then towards the middle, we did upgrade that to the fully integrated card so that everything is kind of nicely packed together. And then on the right hand side, you actually see now, this is a picture from, I think, 2 years ago, where we have the FlowVPE also up and running in our commercial facility, monitoring the processes over there as well. Good. And then, so as said, I think this the last couple of slides just highlighted a bit what are the different aspects that need to be taken care of while implementing a PAT tool such as a FlowVPE. And I think very crucial, or a learning that we really had, was that the very close collaboration that we had with C Technologies was incredibly important to bring that sensor up to a GMP level. So initially in 2016, we did have some say development work to be done on the flow cell and the fibrette in order for them to be cleaned and sterilized adequately enough. And so that very strong collaboration with Repligen allowed to do that in a relatively short amount of time. And I said what we're currently having established is that we're regularly using the FlowVPE system on our clinical and commercial DP batches in various applications, right? But I think the most important application, or the most common application at this point, really is the mixing endpoint during formulation, as well as then or at the end of the dilution process, and then also performing the official IPC at the end of the process. And just in the third and final part, and I also want to do a time check, so I want to probably start wrapping up here as well. But I just wanted to shed some light on what else we can do with FlowVPE and what's the journey that lies ahead of us, right? And maybe just probably a very simple sketch on PAT in general. So PAT is not only, let's say, a sensor that you implement into your process and you collect data and then you transform the data into information for what you're looking for, in this case, protein concentration. But what is also very important is that sometimes we really want to use that information to feed back to the process, right? So to control the process and really step away from that manual, very iterative manufacturing process that we typically have. And so to come back to the example I highlighted before on the dilution process, so when we have the FlowVPE implemented, we're already making the process much more timely. We have a strong lead time reduction. We're removing potential causes for human error, but we do see that there is still the potential to improve that further, right? So how do we see that in the future is that we're going to step away from that 5-step process and we're going to go to a 3-step process, right? So step 1 and 2, they stay the same, but instead of having that sampling in the beginning and then performing a manual calculation and then sampling again at the end when the process has been completed, we're really going to use the FlowVPE in a real-time continuous mode where continuously the FlowVPE is analyzing the concentration during our process, and then with a model predictive algorithm is going to drive that process rather than to have an operator really perform some manual calculations. And so with this, you're really fully utilizing the whole concept of process analytical technology, not only for monitoring, but as said, really to control the whole process, right? So you're really at this point removing the human person, which is the most, let's say, reliable origin of errors sneaking into your process. So removing the human intervention, not having it in the measurement in the QC lab, but also not having it in the manipulation of the process and an interpretation of the data coming from the sensor. So, and then the additional benefit, what you get from that is really much higher control, much more tightly control on the process, and then at the end you end up with a much more robust product quality exiting your facility. So how would that kind of just look like in a very simple sketch is that you have, again here, a specific tank with the FlowVPE interfaced. FlowVPE is sending data into a server. On that server is your model predictive algorithm running, and the algorithm is just sending data back to the manufacturing equipment, and then manufacturing equipment is updating basically your process settings. So all of this, as said, really to replace that human interaction, that human person, which is now really a strongly connected part to the whole manufacturing process. And this is just some example data of the process that we are currently developing, right? So we've done a simple lab-based case study where we are developing and testing out that model predictive algorithm. So here we just went from, let's say, a higher concentrated solution like 90 mg per ml, where we start to introduce the raw material into the tank, and then we, let's say, the algorithm really controls the addition of the buffer into the tank up to the point that you're getting close to your final concentration, and then ultimately finishes the process. And at the end of the process, the FlowVPE would then also be able to perform your official IPC, your in-process control measurement, and then close out the process, and then the operator can continue up to the next stage. So all of this includes and also the automated review and then the data handling. I think that brings me a bit to my last slide, right? So I said next steps, what we're currently going about with the FlowVPE is really to implement that fully controlled manufacturing process step. So what we'll have there is really significant lead time reduction, but also operational flexibility. And then really that automated control allows us to be fully free from human interventions. And then the end goal really is to have a manufacturing process that time after time is executed in exactly the same way, irrespectively of incoming material properties or environmental conditions. A model predictive algorithm should be able to execute the process in the most robust way, such that we end up with the most consistent product quality. And with that said, I do want to thank you again for having bared with me until the end of the presentation. I apologize for going a bit over time, but I hope that there are still a couple of questions we can answer during this webinar. So thank you again, and I'll open up the floor for questions.
Hey, thanks, Raf. It was really great and very informative. Thanks for putting this all together. We do have some questions on the line. I'll read them for you. The first one here, and you kind of touched on it, but maybe you can elaborate. It's mentioned that buffer blank correction is not required. Is it because the path length is very small? Also, what if there is Triton in the buffer protein solution in some amount? And I might be able to help shed some light on that question too, but go for it.
Yeah, no, good question. And let's say no buffer background required might be a slight overstatement in the sense that typically for, I would say 90% of all of the products, we do not need or we do not require a buffer background check. And that's for all of the products that, let's say, are maybe above 1 mg per ml, going up to, let's say, the full limit of FlowVPE, which goes up to 250 mg per ml, where depending on what are really the excipients in your formulation, most of those, the ones that we use in drug product biologics fill finish, they do not contribute to such a big extent to the signal at 280 nanometers. So for those products, the variable path length methodology really allows you to have a certain, let's say, accurate concentration reading without any buffer background. This is something that obviously needs to be tested or evaluated as part of method validation, because there are certain instances where a buffer background might be needed. That's something that we have traditionally seen for the lower concentration range. So if you go below the 1 mg per ml, really, let's say, maybe stretch it out to the 0.1 mg per ml range, where obviously the buffer starts to contribute a bit more to that total signal coming from the protein, where the FlowVPE has also the option to actually perform a buffer background measurement at the beginning of the process that you then can take into consideration. So there is the possibility if it's really needed to, and I would say especially if there are certain components in your system that might attribute to a signal at 280, a buffer background check is indeed required. I don't know, Ramsey, if you want to add to that.
Yeah, no, I think that, that's exactly what I would have said. Well, you even said it better. In fact, you answered the second question here, which is what's the range of protein concentration? I'm going to field that one because you did say it's, about 0.1 to 250. I want to just say that the FlowVPX that's recently come out, has increased that concentration range to much higher. We've done lab studies to measure over 300 mg per mil. And in more anecdotal tests last week, we did some runs up to 170 slope value, which would be equivalent to an antibody at 1100 mg per mil. So I think the future for high concentration is really at the VPX, and that's something that you guys can also look into. Moving on. How much difference was observed between the online FlowVPE and the offline analysis using your qualified method?
Yeah, that's a good question. And always, I still find, we've been been challenged on that over the many years prior to really fully validating our first method, because you always have the question, right? If you're introducing a new method like FlowVPE, you will always have to show comparability to the method you're trying to kick out or trying to replace, which to me puts a bit of a burden, because that means that you can never be better than the method that you're trying to replace, right? So that's just maybe a side note, but we have now kind of merged into a different approach where we are trying to find ways to validate online methods such as FlowVPE without having to do a direct comparison to offline methods like 880. But maybe to directly answer to your question, traditionally we have in the beginning always done a comparability study between FlowVPE and 880, and the acceptance criteria that we have set forth was 97 to 103% across the whole range that we want to validate. So if you have a product and you're diluting it down from 100 to 10 mg per ml, over that range from 10 to 100 mg per ml, for all of the points that you're doing linearity, accuracy, precision, which have to be at least 5, right, according to ICHQ2, on all of those points, you're also comparing to 880, and both data results have to be within, let's say, 3% of each other. And we've been able to validate that for a handful of methods so far.
Okay, so there's a number of other questions here, so we're going to try and get through them all here. So I'm going to skip around on this list, but when we do dilution, do we have control of diluent flow based on mixing time, as there would be some time lag? And associated with, there's another question, actually very similar, that says, what is the sampling time lag relative to the overall process completion time? So maybe you can answer those two at the same time.
Yeah, it's and it's a absolutely great question, because that's the complexity that needs to be built into that model predictive algorithm, right? Because you have components into your mixing tank that are being mixed together, which take a certain amount of time. Let's say that might take a minute for homogeneity to be reached. You're adding your buffer at the same time, which might be at another time scale, and then you're having your recirculation loop, which is extracting material and sending it to the FlowVPE, which, depending on the flow rate in your loop and the length and the diameter, that also takes a certain time. And then you have the analysis time of FlowVPE, right? So you have all of these different mixing times, all of these different time scales that are taking place, that always result into what you're measuring is always too late for what's happening in your process. So especially in a dilution process where you at all costs you want to avoid over dilution, you need to prevent that. And that's characterizing, or being able to characterize all of those different time scales and building then that intelligence into the model predictive algorithm is that's really where that's a very crucial aspect. I think something as simple, maybe as a simple PID control algorithm, might not be sufficient to deal with that. That's where you really need to go into that model predictive control, where you're already knowing the time scales and the lag that you're having compared to the actual process. You can already anticipate to that, and then having the execution of a well-controlled process using FlowVPE.
Yeah, so I'll just reiterate the FlowVPE sample testing time using our standard method is somewhere between 10 and 15 seconds. And then, Raf, do you know what that is compared to the overall process completion time?
So a standard, I'll say, dilution process would take, without all of the waiting times, right, in between, so just pure execution, would take, let's say, an hour and a half in total.
Cool.
Currently, which is obviously something that we want to reduce significantly with FlowVPE.
Great. So how do you perform the FlowVPE system suitability test in an inline or online GMP environment?
So as part of the system suitability test, there are a couple of things that you want to check out, right? So on the one hand, you need to be able to check whether the equipment on itself, aside from the flow cell, so the direct product contact parts, you want to make sure that that's okay, and that's something you can typically do with a reference solution like the Chem 13 or the confirmed standard. You can also do a typical transmission test to see if the optical fiber cable is working properly. So that's really tests you can do on the equipment prior to production, not utilizing any of the components that will come into contact with the final product, because they might get contaminated by the reference solution, for instance. But then you do also want to do some testing on the actual flow cell and fibrate you're going to use into your production, because they might have been damaged by the cleaning or the autoclave process, or an operator might have dropped it on the floor, right? So to that extent, we have established also a couple of procedures where we do, say, an alternative version to the transmission test with the flow cell and fibrate mounted in their final position, and we also do another say test to verify for cleanliness of the system, doing let's say a standard measurement with an empty flow cell. So combining these will allow you to determine that the whole instrument is working correctly, but then also the components you're actually bringing into contact with the product is not damaged because of the cleaning or the sterilization processes prior to production.
Great. So yeah, we're kind of at time, but there are a couple of more interesting questions here. I hope people can stick around for these. So hi, are you using this as a platform, or do you only use it for certain product lines? And if so, why? He says, I know quality sometimes wants process-specific PAT validation.
So FlowVPE currently is not a platform method at this point yet, but we are in the process of really onboarding more and more products now over the last 2 years that are utilizing FlowVPE as part of the dilution process. But it's not really that it's a platform process at this point, because we typically take the approach that we onboard FlowVPE for new products that we introduce into the facility from a regulatory filing burden perspective. We don't want to update filings unless we have to. So to that extent, all of the new products they are coming into the facility, they are being equipped with FlowVPE in the process. And now you have to help me, Ramsey, what it was the first part of the question. It escapes me right now.
Well, it was just, are you using it as a platform method, or do you have it on certain product lines? I think the answer is right now you're using it for certain product lines, mainly due to filing concerns for the previous products, right?
Yes, correct. Yes.
Cool. And then this is the last question we have here is, most of the data you showed was at 5 mg per mil. This is quite a low concentration based on recent industry trends. You showed a little bit of the 90 mg per mil data at the end of your presentation. Any insight into the upper concentration limit, which I think we've kind of discussed, but maybe you can specifically talk again about the accuracy and precision of that high concentration limit that you've tested.
Yeah, and maybe you can speak to that as well, Ramsey, because I mean, for drug products specifically, most of the products that we do fill are, let's say, in the concentration range from 1 up to maybe 120, 130 mg per ml, and in that range, and I've not shown all of the data obviously, but we have multiple data to show that in that range we do have, let's say, the possibility to validate the analytical method. So again, here, plus minus 3% to the offline 880. For higher concentrations, which is something that would typically be seen on the drug substance side, we are also looking here into several products. But then to Ramsey's point before as well, is that FlowVPX really has a lot of benefit to go really up to incredibly high concentrations, which we are indeed seeing coming down the portfolio, where we're talking about 300, 350 mg per ml concentrations coming down the pipeline. And so up to this point with FlowVPE, we're still waiting for us to test such high concentrations, but it's on the horizon that we will do this in the near future.
Yeah, and I can add a little more color there. I would recommend going back to Jay West's presentation from last week, where he showed some data up to about 260 mg per mil for a somewhat higher extinction coefficient. And hopefully I can at some point show the work I did last week, where we did a 85 mg per mil oligonucleotide, which if anyone here is familiar with oligos, they have quite a lot of absorbance, quite a high extinction coefficient. So I think the capability of the VPX was demonstrated there at the high concentration. And there's no reason from a technical perspective, in my opinion, why the accuracy repeatability number should be any different, because the method is consistent across the range as long as you're within the linear range of the instrument and you're getting a high R-squared value for the instrument. So I'd be happy offline to talk about that some more, and also even provide some more data to show anyone who wants to see that stuff. So with that, we're just a few minutes over time, which is, I hope, not a problem. If there's any more questions, please pop them in right now, and we'll ask them. Otherwise, thanks for everyone for joining. Thanks, Raf, for the presentation. It's been great. And also, just please remember, there's one more in the series, which will be our panel discussion with Tony Hunt next week, next Tuesday at the same time. Please join, and hopefully we'll have some more interesting topics to talk about as a group on the final Go with the Flow expert webinar series. So thanks a lot, everyone, and I hope to see you next week.
Thank you very much, everyone. Have a nice rest of the day, and stay safe.